Hey everyone, so today we're going to continue our look at the biological perspective by looking at techniques used to study the brain. And this is really a topic that gets us back into thinking about how do we learn more about the brain? What technology is available to us to really help deepen our understanding of the most important part of our body, the brain? So this first video, and there's one of two that I want you guys to look at in the PowerPoint version of this video. But the first one is just a really cool video that looks at uh, some neuroscientists that actually made brain imaging into art. And so there's certain things like you get to see the path that a neural pathway takes. And it really just illuminates for us the beauty of our brains in terms of how they work, in terms of really, what they're producing for us, allowing us to go through every single action that we have as a person. Now, getting into techniques used to study the brain, there's a few different factors that lead a scientist or researcher to choose a particular type of technology. And the first of these is opportunity. Um, the second one is available technology. Is this able to be brought to them? But the most important sometimes is cost. And I'm probably underrating the influence of cost, but oftentimes, based on the technology, it can produce a cost for the research that is substantially higher than other technologies. And so, unfortunately, that does become a choice when considering the possible scientific advantages of using other possible technologies. The other important factor here is really based on what the researcher wants to know as well. As we'll talk about, there's certain benefits and drawbacks of using each of these technologies. So the first class of brain scan techniques we're going to be looking at here study electrical activity. And this is really a non-invasive way to study the brain. And it's good for studying language development in babies and small children because they can't necessarily talk, sleep stages because not all of us talk in our sleep, or at least coherently talk in our sleep, and then also neuroplasticity. So seeing over time the changes that occur within the brain due to a certain environmental factor, learning, understanding, growth, something like that. So the first of these electrical technologies is EEG. It is an electroencephalogram, and you see an example of that in the picture in the right corner. Um, those are diodes all over the person's skull. And this is good for just understanding general brain activity. So it's especially good for studying babies. They don't need to speak in order to be measured. And the tissue from the scalp and skull are passive conductors of electrical current. And it gives a constant reading and recording of general brain activity. It's also good for sleep studies. So for example, the stages of sleep, we know someone is asleep during stage 2 when a burst of sleep spindles appear on the EEG recording. And the spindles are these little lines right here, which tell us the nature of someone's sleep. However, there are a few limitations. The first one is that it's not good for studying the brain when a researcher wants to know which brain regions are activated during cognitive tasks. And it's also somewhat sensitive to movement. And so you have to stay still. And so what that does is it sort of limits the types of research that can be done with EEG. A couple of other electrical activity techniques. There's a MEG a magnetoencephalography. So uses sensors within a MEG helmet to record the small magnetic fields produced by electrical currents in the brain, and it allows for a precise localization of the neural currents responsible. So you can actually see where the currents are occurring within the brain. The other one are TMS, transcranial magnetic stimulation. So this is a sort of they put electrical currents through a well-insulated copper coil, and it produces a magnetic field over a specific place on the head. And a TMS coil, they sort of take this wand and put it over certain parts of the brain, and that lack of power within that part of the brain can sort of show scientists what exactly that brain part does. If you put the TMS coil over a certain part of the brain and the left arm starts spasming, that tells you a lot about what that particular part of the brain does. So it's been quite helpful in certain scientific research opportunities. All right, today we're going to be talking about two types of imaging though. Before we talked about electrical brain scans, but now we're going to be talking more about pictures. So there's two types of imaging, structural and functional. And we're going to talk about structural first here. So structural imaging measures physical properties in the brain. It allows for greater visual imaging of the specific areas of the brain, and it also is used in the diagnosis of brain tumors and injuries. And just like with the electrical ones, these are several different types of brain scans that are part of the structural imaging part. So the first one is MRI. I'm sure some of you have had an MRI, unfortunately, for broken bone or some sort of imaging they needed to do on your body. But it can be quite helpful in brain tissue research. It stands for magnetic resonance imaging, and it works by acting on magnetic properties of the atoms in the brain. And that's why you have to go into that giant tube, because they are using these magnetic properties. And so this becomes useful when a researcher wants to study something structural about the brain. How does something look within the brain? How does the structure of it work? But it is expensive. It is time consuming. Some people have to go into an MRI for 45 minutes to an hour. If you're studying thousands of subjects, this may not necessarily be good. But also all equipment must be removed from the MRI room when the machine is in use. So what that means is that you can't necessarily have someone, doing a puzzle to see what their memory is like or how their brain is working during the use of this puzzle. And so it becomes quite limited in scope because of that. So the first study we're going to look at today actually used an MRI, and this is Henry Molaison. This is Scoville, Milner, and Corkin. We've talked about HM several times at this point, but this is a case study with brain scans, and they wanted to understand his antegrade amnesia, and so amnesia that occurred after his surgery to cure his seizures. And what they did was that part of Corkin's research later on, an MRI in 1997 showed that the cerebellum had atrophied. And so what it also showed was that the hippocampus damage wasn't quite as extensive laterally as first thought. So nearly 45 years into research on HM, they have discovered more from brain scan techniques. And so if you were to write an essay on HM, or be asked a question, I should say, about brain scan techniques, and you used HM in your explanation, then this would be what you want to focus on. The fact that this 1997 MRI had shown that the hippocampus damage wasn't quite as extensive laterally as first thought, and it demonstrated more specifically that specific areas of the hippocampus, like Brodmann 38, is what made his memory so bad. And so it gave us more of a picture of the localized role of the hippocampus in his memory and specific parts of the hippocampus as well. All right, we're going to skip past this slide, but it is important for you to know. He also had an MRI in 2008 when he died, unfortunately. All right, this is a good ethics article for thinking about the ethics of studying someone after their death using these brain scan techniques. So I would suggest clicking on the link in the PowerPoint and reading that particular article. All right, the next type of structural imaging are CAT scans. This stands for computerized tomography, and it builds up a picture of the brain based on the differential absorption of X-rays. And so the subject lies in something that looks like that. It's much smaller than the MRI machine, and an X-ray source rides on a ring around the inside of the tube with its beam aimed at the subject's head. There's a few strengths of why a psychological researcher would use a CAT scan machine. There's a rapid acquisition of images. There's a wealth of clear and specific information, and you can view a large part of the body. However, the X-ray also has unintended consequences. It exposes the subject to possible radiation. It requires the patient to lie still, so they can't be completing a task while they're doing it. And so that limits the scope of using CAT scans. Like it says in the title there, CT scans is another name for CAT scans. They also reveal the gross features of the brain, but they do not sort of resolve its structure well. They do not sort of show the structure more specifically. All right, the next classification of imaging, I'm sorry for the pause there, are functional imaging. So this includes fMRIs. An fMRI, like it says there, functional magnetic resonance imaging. So this measures the metabolic changes correlated with neural activity. And a lot of times we'll see throughout the year that fMRI becomes the most widely used brain imaging type because what you can see is that the changes that are taking place as someone completes a task. And so it becomes most beneficial to the researchers. And so the researchers can then have a true sense of cause and effect by having a control condition or a baseline scan, and then compare it with the bold areas imaged during a task to identify brain changes. It also doesn't use radiation like an X-ray. However, it's very expensive. It's difficult to interpret. It is an indirect way to study neural activity because it can't pinpoint specific functions that might be told by individual neurons. And that fact of that researchers still aren't completely sure how it works also influences possible limitations that we can learn later on about the limitations of the fMRI. So that's going to lead us into Carrerias et al. We know by this picture we're talking about the whistled language study. But in Carrerias et al. 2005, which we'll see several times throughout this unit, the aim of this was to discover whether the whistle language was also processed in the left temporal and inferior frontal lobes, and to see whether the brains of SILBO users, the whistled language, differed from the brains of non-SILBO users. So they used 2 scans of fMRIs within this, and you'd want to make sure that you're talking again about the brain scan techniques if asked a question about brain scan techniques. But this is the baseline scan showed them listening passively to SILBO in Spanish, And then the active attention one listened actively to Silbo and Spanish sentences mixed with silent periods. So the results of this, these baseline scans and the the active scans showed that Silbo whistlers in both tasks used parts of the left hemisphere associated with speech production. Those who were using both Spanish and Silbo showed an activation of the right hemisphere. And so this taught us a lot about the possible role that language plays in the right hemisphere, which was originally thought to only be a left hemisphere of the brain process. And so fMRI and its inherent strengths and weaknesses became a strength and weakness of the study, but really helped us to gain knowledge about the workings of the brain. Another type of functional imaging are PET scans. They measure levels of the sugar glucose in the brain in order to illustrate where neural firing is taking place. Now, neurons use glucose as fuel, and so that's why this particular thing works. But as part of the scan, a tracer substance is injected into the blood, and when parts of the brain become active, blood with the tracer is sent to deliver oxygen, and so it creates these visible spots like we can see in this particular look right here. Obviously the blue is passive areas, and the colored areas, or green, red, yellow, are all areas where brain activity is taking place. So a few strengths of this is that a video image of the brain is being used here and can be made while the subject is performing a particular task. And so it enables researchers to see a moving image as well as the difference in brain areas during the production of a task. Again, a lot of psychological research, especially the strongest ones, come down to an actual causal relationship. The more that we can learn from what causes one thing to occur and affects on another thing, the better. And so imaging that follows this is the most helpful. Limitations: it can only locate generalized areas of brain activity, not specific locations. It's also costly and invasive. And the invasiveness comes from the fact that you are injecting someone with this chemical in order to study them. And so it makes the use limited in that way. So the last thing I want you guys to look at here is a TED Talk where they talk about the most important lesson of brain scans, and they've looked at 10,000 brain scans and to see how they have sort of changed the way we look at the brain, but also influenced psychological research. All right, that's the end of our talk today. If you have any questions, please email me or find me in class. Again, brain scan techniques are a powerful tool of psychological research, even if many of them have their own inherent strengths and limitations, which may change the course of when they are used.